Hello, everyone, and welcome to today's webinar. Before we begin the webinar, let's take a moment and allow everyone to settle in. All right, so let's get started. My name is Sanchita Bhattacharya. I am the Scientific Outreach Program Lead for ImmPort. It's a pleasure to have you all join us for this insightful session. And today I have ImmPort members with me, Kate Hypes, Sammy Smith, and Ruben Serval, and they will be helping me throughout the process. So before we go to the main presentation, I would like to cover a few housekeeping items. The presentation time is 45 minutes, followed by 10 minutes for Q&A. And throughout the session, we encourage you to ask questions using the Q&A feature. And we'll try to take a few questions live during the Q&A segment. And this session is being recorded and a copy will be made available for reference later. And I'm going to provide a 5-minute introduction to ImmPort before we begin the main talk. So many of you may already be familiar with ImmPort. Either you have submitted data or downloaded data sets from your own research or explored the platform out of curiosity. But before we dive into today's main presentation, I would like to take just a couple of minutes to give a brief overview of ImmPort. So ImmPort is a data portal which is developed to support NIAID DATE-funded researchers by providing a secure structured platform to collect, share, and reuse immunology and clinical trials data. ImmPort ecosystem consists of private data where investigators can upload their data using guided templates, and this area supports data entry and internal management prior to public release. Shared data is a public access to standardized data sets. Users can explore immunology studies across diverse domains. Data analysis, we have tools and some workflows for analysis. And then resources, we have a comprehensive documentations and tutorials for data analysis that is available to the community. ImmPort aligns with FAIR principles to promote findable, accessible, interoperable, and reusable data. And then also I would like to mention that ImmPort is not just a repository. It is also now enabling the AI-ready, reproducible, collaborative science. So please do visit immPort.org if you are interested in accessing the curated data. So many of the data users, they ask, where is this data coming from? So just to let you guys know that ImmPort supports NIH data sharing mission by serving as a central repository, and all the data is coming from NIAID programs, and as well as external organizations are also submitting data to ImmPort. The list is long, but I would just name a few of them here, like HIPSI, Human Immunology Project Consortium, Seronet, and IMPACT for COVID-19 data sets. We also have some data sets from Bill and Melinda Gates Foundations, March of Dimes, and Civics program. And in the broader research domain, such as vaccine development, allergy, and asthma research, transplant, autoimmune disease, we have a variety of data sets which are getting submitted through consortia, big programs, as well as small research study programs to ImmPort. And ImmPort also powers public access to clinical trials data. And ImmPort is also recognized by Nature Scientific Data +1 and listed in the re3data.org as a trusted and fair aligned repository. How do we access ImmPort data? So the shared data browser in ImmPort is an interactive cohort discovery tool designed to help researchers easily explore and filter publicly available data sets. It provides a transparent, structured access to a vast range of data shared by the projects. As of May 2025, and we do monthly release of the data sets, ImmPort hosts over 1,200 studies for more than 175,000 subjects, data across 176 diseases, including 185 clinical trial data sets. We have some rich filtering options, and as you can see, there are faceted search. You can filter the studies by the research areas, condition or disease, assay methods, demographics, study metadata, and so on. Users can download these studies, export the filter results, and even use the API for programmatic access. This tool supports fair data use and also has been helping with the AI development and reproducibility checks. So I will highly encourage people to go to ImmPort website, immPort.org, to explore data sets. So we have just seen how ImmPort presents opportunities for researchers to explore and access rich data sets through this shared data browser. And now let's shift gears and highlight an exciting example of how those data are being put into action. With that, it's time to turn it over to David Ding. David is a postdoctoral fellow in Dr. Butte's lab at UCSF. Today, he is going to present Auto-MedCalc, automated biomarkers discovery and risk generations with AI agents. So with that, I'll turn it over to David.
Thank you, Sanchita.
Let me stop sharing.
Hi, everyone. I'm David Ding, and today I will give a talk titled in AutoMedCalc, Automated Biomarkers Discovery and Risk Score Generation with the AI Agents. So let's take a brief look on the studies on ImmPort. So ImmPort is as we have introduced, is an open, public, and large-scale data platform for immunology. And in this platform, we have a lot of studies. For example, we have more than 1,000 studies and a lot of subjects across different species, and we have a lot of experimental results. So such a huge amount of data we have, public data we have in the immunology fields, and it really motivates us to use some AI models in this data-driven way. So today we will have a specific focus on a case study in organ transplant, because organ transplant is also a very important topic in the field of immunology. And here I have a high-level illustration of the organ transplant allocation and the post-organ transplant management. So the organ transplants, one of the main tasks of organ transplant and one of the most important essential one is the allocating the organs, because compared to the patients on the waiting list who need the organ is a very scarce medical resource. So how to allocate these organs to the patients waiting on the waiting list? So usually we will calculate a kind of score for them. For example, we will have the MELD score, the MELD score for the liver transplant, and then these patients will be ranked with their priority. And we will also take other factors into the consideration when we do this kind of organ allocation. For example, the distance between the organ and the recipient, and also the waiting time of these patients already on the waiting list and the age of them. And based on these factors and their ranking of the patients, the doctors will make their allocation decision. So after the transplant allocation, we will conduct a surgery, and then another important task is the risk evaluation. So we need to evaluate what kind of post-transplant complications this patient may suffer from. For example, the rejection, the mortality, and the infection after the transplant. So from this example on organ transplant, we can find that it has a specific needs and an urgent need for the medical score calculation. We need to calculate some score to rank the patients priority to receive these organs, for example, based on their severity. And also we need to evaluate their risk of suffering from the rejection or infection. So from this example, we can find that the medical score calculation is a very essential step in the clinical practice and biomedicine research because we always need to calculate some score to help us diagnose and to identify something. And here, let's take a look at the current medical calculator. So what is the traditional way and what is the widely used way in the clinical practice? So on the left part, I put a figure. It's a screenshot of the open source. It's a very useful and open platform with more than 450 medical calculators designed by the human experts, by clinicians, by researchers, and for different type of disease. However, it's a very useful platform that we can search in for different type of expert designed medical calculators, but there's still some limitations of this kind of human expert designed rules. For example, we summarize the two limitations here. The first limitation is the very weak adaptability to different medical scenarios. For example, we have so many different diseases. And for even the same type of disease, we have so many different kind of conditions. So it is very hard to design a specific score that can be adapted to different type of disease. For example, we can design a score for to evaluate the kidney function, like a EGFR, but it's very hard to adapt this EGFR to other type of disease. And the second limitation is this kind of traditional medical diagnosis using this kind of scoring rules, it is very hard to identify the new biomarkers and the important features because it's an heuristic way. So which means we will usually design this kind of input features based on our already known the knowledge and the expertise and the experience. And then we're using this fixed group of input features to calculate something, to decide something. So this kind of traditional way actually blocks out the chance to find the new biomarkers. However, we have so many data as we have seen, for example, in the ImmPort, we have so many data. So we may ask, can we have a data-driven way to identify the new biomarkers? And also here I put two examples here to show some limitations of the expert designed score in a heuristic way. For example, the MELD score. So MELD score is a clinical widely used in liver allocation. But from our preliminary experiments, we find that the MELD score has very weak indicative capability to indicate the post-liver transplant outcomes, for example, like the infection or mortality. And the second score is the impact score. So impact score is a score specially designed for heart transplant, but it cannot identify new biomarkers from blood and from urine. And this is also common challenges for many other heuristic design scores. Then let's take a look at something. The new paradigm. So it's building agentic AI for procedural automation. So as we have introduced examples that we have a motivation that this kind of conventional scoring in clinical and biomedicine is a complex process, but a very logical one. So usually we'll first identify essential biomarkers or features and then design some kind of scoring rules or using some kind of algorithm like a logistic regression, and then we output the results. It can be some outcomes like a mortality, diagnosis, and then we analyze these statistical results. So it's a very logical way. And so here we come up with some new paradigms. So can we develop a fully automated pipeline to automatically this process, to automate this process? So here we introduce the agentic AI. So what is the agentic AI? So agentic AI is autonomous systems that can independently pursue the goals and complete the task in an automated way. So maybe this kind of agentic AI could help us with this process to automate the process. Then let's take a look at a detailed look at our framework. And this is it's a multi-agent system for medical calculation. So first step is we will have a lot of input variables. For example, we have clinical parameters, we have molecular data, we have medical history, we have demographic information of the patients, and also history of the medical history of the patients, and we have all these kind of input features or variables. And this kind of raw data will be input into our agent system without many pre-processing. We don't have to do many feature engineering upon this kind of raw inputs. And then it's our Auto-MedCalc agent system, which consists multiple agents. So we call it a multi-agent system. and they will have several discussion between them, and we will introduce the details of how we're building this agent in the next few slides. So the second step is the agent. One of the biggest, I think it's a difference between the agent and the pure large language model is the agent, they are be able to call or will be able to use different type of tools. For example, computational tools and the statistical analysis tools, any kind of tools that can be used by this agent system. For example, in our framework, our Auto-MedCalc agent can use multiple tools. For example, machine learning model, it can call the large language model, and also light GBM, and also it can call the feature importance calculator. It can calculate which feature has higher importance in this diagnosis or in this score calculation. So it will call or use different type of tools by the API. And the third step is it will directly output the results for us. So the agents will identify salient biomarkers and predict the risk score, for example, the rejection or the mortality. So from this high-level illustrative figure of our AutoMedCalc agent system, we can find that the agent can take the input like raw data and then code using different type of tools and automatically and complete our assigned task to them. For example, diagnose the rejection after transplant. And then let's take a more detailed look at how to build the agents and how to let this agent collaborate with each other. So which is also our core technical contribution in this work. So the first step is how to build the agent. So we're building the AI agent with from the large language model. So for example, we have two agents built or derived from large language model in our framework. So the first one is the machine learning engineer agent. So it's an agent derived from the large language model by prompt learning, by prompting. So for example, we can prompt to the large language model that you are a machine learning engineer, and your goal is to help us building up the machine learning models, select the important features, and also test this model and optimize the input to achieve the optimal performance. And similarly, we derive our implement our second agent, which is the domain expert agent from another large language model, also by the prompt learning. So we can prompt into the large language model that you are a domain expert, and for example, you are a transplant expert, and your goal is to help us to make sure that the input features used for our machine learning model make sense from a medical standpoint, make sense from a transplant standpoint. And after we have building these two agents, we need to let this agent collaborate with each other. So they are two independent agents from two large language models. So how to let these two agents collaborate with each other to improve the performance, to achieve better performance. So here we have a paradigm or topology of this kind of collaboration between two agents by the discussion. So these two agents, they will have several rounds of the discussion until a consensus is reached. For example, the machine learning engineer will prompting, and this is the added and the deleted features, and it can improve the performance, it can improve the accuracy. And then our domain expert agent will determine, okay, whether these selected features by the machine learning agent make sense in a medical perspective, whether the selected features are really related to our transplant outcomes, for example, the rejection, are they really related from a transplant expertise standpoint? So after we have this building and the collaboration of the two agents, we can building the whole agent system, and they can automatically help us complete this diagnosis task. And then let's talk more about the materials and why we use and why and the motivation that why we choose the transplant as our research example. So we think transplant is a very good representative research example to conduct experiments to use as a test bed. So we choose it for several reasons. The first one is there is a critical unmet need in transplantation for reliable tools to score and predict the clinical outcomes. For example, we need to score the patients to see their severity and to see their urgency to receive the organs when they before they receive the transplant. And after transplant, we need to predict their rejection and infection rate. And the second point is significant efforts are focused in this domain are focused on identifying biomarkers that can accurately predict transplant outcomes. For example, in the transplant community, there are a lot of efforts put into identify and discover the new, especially the non-invasive biomarkers to quickly and precisely identify the rejection after transplant. So in this work, we focus on the rejection after kidney, liver, and heart transplants, these three main types of the solid organ transplants. And here, before we go into more details of the data we use, here is the data that we used comes from it's a clinical trials in organ transplantation, or CTOT in short. So the three studies, the study 557, study 1403, and study 571 are all the studies that conducted under the CTOT clinical trials. And we can take a look at their original research goals from these studies that we use from the ImmPort. So we choose the three studies of organ transplant from the ImmPort, which are kidney, liver, and heart transplant. So for the kidney transplant, we have 208 patients for this original study, and their original goal is to assess the predictive value of the non-invasive monitoring test from urine and blood as correlates with the kidney post-transplant outcomes. And for the liver transplant study, it's the study 1503. And in this study, they have 203 patients. And the goal of this study is to develop blood and or urine tests that will help to detect early signs of the rejection in people who have had a liver transplant. And the third study is the heart transplant study. It's study 571. It contains 200 patients. And the goal is to determine whether any single monitoring test or combination from urine and blood is associated with the primary endpoint. So we can, from these three original studies and their original research goals, we can find that most of the studies, they have a specific focus, and the goal is on the identify these biomarkers from the non-invasive monitoring tests, for example, from the urine, from the blood, and to early detect the rejection after transplant. And here is how we aggregate in these transplant studies. So because these studies are under the same clinical trials program, it's a CTOT, so we integrate these multiple transplant studies to build an AI-ready datasets. And for example, we can find that this kind of studies, although some of the study, they are conducted by different groups and they have different specific focus. Some are on kidney, some are on the liver, but they share some common features. For example, the age, the ethnic height, weight, this kind of common features are shared between these kind of studies. So it provides a very good opportunity to analyze these studies, these different studies simultaneously, and connect them with each other. And here's the transplant clinical trial data dictionary report, and it's more detailed example of how these two studies can be put together, and we can analyze some common features between them. For example, in the study 557 and the study 571, there are kidney transplant and the heart transplant respectively, and we can find that they share some common features like RBC, the red blood cells, both are tested in these two different studies. So here is more detailed information of our of the transplant studies, the three transplant studies that we use to build our model and test our model. So we have three studies, and they have in each study they have different type of assay types. For example, ELISA, mRNA expression, chemokine, and they collect the samples from different type of sources like urine, blood vital signs like blood pressure. So we have we can find that from the for the input of our model, there's a mixture of this type of data. So their data comes from different sources and by different assay types. And then let's take a look at the what does the data look like. So what is the data look like? So it's actually a mixture of data types in these studies. So we have some structured tables, structured tabular data, for example, demographic information of the patients, the medical history of the patients, this kind of static information with the patient. And then we also have some temporal data or time series data because we in many lab tests we collect this kind of lab test in a longitudinal way. For example, we have a transplant date for this patient, and then they will collect the lab test at a different time stamp. For example, at day 1, day 5, day 10, to collect in this kind of time series way. So we also have this kind of time series data and also a mixture of structured tabular data. So then let's take a look at our models implementation details, how we implement our models and the baselines. So we use a fivefold cross-validation method to do the cross-validation. And the baseline we use, there are 3 categories of the baseline. So the first one is we will compare our method, the agent system, AutoMedCalc agent system, with the human designed rules, which are specifically expert designed rules for this kind of organ transplant. And then we will compare with the baseline machine learning models, for example, the light GBM, MLP, random forest, and the logistic regression. And the third one is the baseline feature selection method, for example, threshold, univariate, tree select, this kind of feature selection methods. And we use the metrics of ROC-AUC, the area under the receiver operating characteristic curve, to evaluate the performance, the accuracy of our models and the baseline model. So here we have a more introduction of the human designed scoring rules. And these scoring rules, they are commonly used from the literature and also the real world clinical practice. For example, for the liver transplant, for the kidney transplant, we have the human baselines, including the EPTS and the KDPI and the EGFR and the ratio between the blood urea nitrogen and the creatinine, and also the UPCR. So we're using this kind of commonly used kidney function or kidney disease evaluate score to evaluate and predict the rejection after kidney transplant. And the second type is the heart transplant score. It's a heart transplant human baseline. For example, the transplant risk score, TRS, index for mortality prediction after cardiac transplant, the impact score, and also risk stratification score, the RSS, for heart transplant. And for the liver transplant, we have also 2 commonly used human baselines, the MELD and the MELD-NI, the MELD sodium score. And then let's take a look at our results. So it's the transplant outcome prediction performance, and specifically it's on rejection diagnosis or rejection prediction after the transplant. So let's first take a look at the results on the kidney transplant. So here we make the comparisons against the 3 categories of the baseline, as we have introduced is the human design rules, baseline machine learning models, and the feature selection method. So we can find that for the human designed score rules, and this kind of human expert designed scores for kidney related disease didn't has a very precise indicative performance of the rejection. And from our these preliminary experiments, we can find that the AutoMedCalc can precisely predict the kidney rejection after the transplant. And here also we can find an interesting thing here is this kind of kidney design, human expert design scores for kidney transplant. They are not specifically designed for kidney transplant. Some are used for widely the kidney function evaluation. For example, the EGFR, and so we can from our studies and our data and from these preliminary experiments, we can find that the EGFR has a very weak adaptability to this kind of kidney transplant prediction. And also, we make our models comparison against the baseline machine learning methods. For example, the light GBM, which usually performs very good in this kind of tabular data and time series data. And we can find that the AutoMedCalc can outperform this kind of baseline machine learning models. And it also makes sense. So the reason behind this kind of better performance from our model is mainly due to our model can select better features compared to the baseline machine learning models, for which we input all the features into these models. And we add one more step by our agent, and we select these features to select the most important and the most salient features and the most relevant features to as the input. And from the third figure on the right part is the comparison between the feature selection method, for example, the threshold, tree select, and the univariate. So we want to show from these results that how could our agent system select these features from the raw input compared to the traditional and usually the statistical based feature selection method or pure computational based feature selection method. And we can find that it has better performance than this kind of pure statistical or computational based feature selection method, because our agent system it selects the feature not only based on this kind of computational analysis. For example, we only select the top 10 most important features. Our agent system, it can also analyze the semantic meanings of each feature, because we have the feature name as the input to our agent system. So the agent can it is derived from the large language model, so it can has the knowledge, has the already known knowledge for which feature can be more related to the kidney rejection. So it can do this kind of selection based on the semantic meaning of each feature's name. So that's one reason that contribute to why our performance is better than some traditional based feature selection method. And here we also have the results for the liver transplant. So we can find that compared to the MELD NA, MELD sodium and MELD the original MELD score, so the AutoMedCalc agent can also achieve better performance in the liver rejection diagnosis. And from this figure, I think and very interesting here is we can find that the MELD NA, the MELD sodium score, it actually has a competitive performance of the liver rejection diagnosis. For example, it achieves the 72 ROC AUC score compared to our 89 from our agent system is still very competitive. So we want to show from this example that although the agent system or some powerful AI models can still outperform the human expert design rules, but in some circumstances, and the MELD the human design score like the MELD score, it can still have very good performance. It can still deliver very good and precise diagnosis results. So from this example, the MELD score, we think it still can work. So that's one example that how could we combine this kind of agent system and also our human expert design the rules in this kind of task. And also, from compared to the baseline machine learning models, we can find that in the liver rejection diagnosis, our agent can still outperform the light GBM, logistic regression, this kind of baseline machine learning models with better input optimization on the features. And also, we can outperform the feature selection method that are purely based on the computational analysis. And here is the heart transplant results on the heart rejection diagnosis. So similarly, we can find that like similar results as the previous two slides, that the agent system can precisely identify the rejection after heart transplant compared with the human design rules and baseline machine learning models and the feature selection method. And in this figure, in this slide, we want to show more that our agent system, the AutoMedCalc, is not a black box system that as we have introduced, it has 3 steps in high level, input the data, and it can use different tools and output the results. It's actually a white box one, it's not a black box one. So we can understand why our agent make this kind of decision in the feature selection to identify these biomarkers. So in this slide, we showed AI agents assisted decision-making process by one example. So we can find that our agent system, our 2 agent in our multi-agent system, the first one is the machine learning engineer. It will provide, okay, which feature to be added and which feature to be removed. And it tested that this can improve the performance of our model. It can improve the diagnosis precision. And then our domain expert will determine whether these features to be added are reasonable whether this kind of feature are really related to the rejection, whether it should be added in our input features. And also it can determine whether the deleted biomarkers, whether they are reasonable to be deleted. For example, maybe some features or some biomarkers, they are not that related, they're not strongly related to our kidney transplant rejection. So it may decrease the performance. So we don't need to include them. So this kind of domain expert will help us determine from a medical standpoint to make sure that every feature is included and they make sense and they are really related to our organ transplant outcomes. And then it will outperforms whether it agree yes or disagree no. So and this is one round of the discussion between our two agent system. And then let's take a look at more results on the important features that we have identified for the rejection. So in this slide, we show the important features that we have identified for the kidney transplant rejection. So let me spend more time to explain this figure on the left. So we plot the Shapley value figure, which means that we want to know which feature are more important to the rejection after the kidney transplant. And the red color indicates the higher value of this feature and of the original features value, and the blue color indicates the low value of the features. And then we can find on the x-axis that the Shapley value is the impact on the model output. So on the left part is the minus part. It indicates that this kind of it indicates it has the negative impact on the rejection, or you can say it relates to the non-rejection or no rejection. And on the right part, it is related to the higher risk of rejection. So we can, for example, from the red blood cell, we can find that most of the high value of the red blood cell that contributes to an non-rejection. And also we can find that the other factors here as well, like the CCL5 is a test from the urine mRNA, and the CXCL5 is also an urine mRNA test. So from this figure, we can find these important features and how could they impact our kidney transplant rejection. And also we have found evidence for these each features with previous scientific literature that we have validated from our previous results from the wet lab and from the clinical trials, that these biomarkers or these important features, they are really contributing to the results of the kidney transplant, the rejection. And here is the results on the liver transplant rejection. So we can find that on the left figure, and for example, the most important feature is the serum potassium, and we can find that most of the low value of the serum potassium that contributes to the rejection after the liver transplant, and for the serum sodium, creatinine, albumin, these several features, they are highly related to the liver transplant. And especially an interesting thing here is we can remember the MELD score. So MELD score is actually calculated based on these important features as well, including the sodium, the creatinine. So it's a very good, and you can also find that these three features, they are the top features, top important features that we have identified for the liver rejection. So this also explains why we can find from our preliminary experiments that the MELD score or the MELD sodium score, it has a very good indicative performance of the liver rejection. So it has, we can find some reasons in these slides. And here we also identified evidence of these important features in the previous scientific literature. For example, this acute potassium and sodium, how it affects the liver's rejection after transplant. And in this slide, we show the results of the heart transplant rejection, important features that we identified from our Auto-MedCalc agent. So we can find that the white blood cell, the BK virus, and some blood test biomarkers, they are very important to increase the risk of the rejection. So and also the blood pressure, this kind of vital signal features that also are related to the heart transplant rejection, to identify this heart transplant rejection as the risk factors and the biomarkers. And we also find the evidence for some of these biomarkers and features we identified from our in this data-driven way, and they has also been validated by the previous scientific literature conducted in the wet lab and in the clinical trials. So in the last, I will have some takeaway messages for this work. So we successfully demonstrated the transplant outcome prediction using the human design rules and machine learning models and the feature selection methods. And also, we find that Auto-MedCalc leverages AI agents to assist decision making and provide insight into the clinical outcome predictions from large transplant data sets. And the third point is the important features identified by AutoMedCalc align well with the previously reported findings in the scientific literature. And the fourth point is our framework shows promise for broader applications. So currently we have a specific case study on the organ transplant, and also we can extend this framework to other immunology related disease and also other non-immunology related disease or conditions that has a need to diagnose and also the biomarker identification. Yeah, and thank you, everyone. Thank you for listening. Thank you. Feel free to ask me any questions.
Yes, an excellent presentation, and this is a demonstration of how open access data sets can be put into action in the context of AI. We do have questions under Q&A, and I'm going to read it in the order we received it. So I'm going to read it loud. How does the multi-agent systems handle batch effects between data sets or within data sets? For example, submitting the mRNA profiling data.
Okay, let me see the question. How does the multi-agent system handle batch effects between datasets or within datasets? Submitting raw mRNA profiling. Okay, I see. Yeah, that's a very good question. So, firstly, so we conduct, because we have three organ types of the data set. So it's like a more three sub data set in a large data set, and we conduct experiments within the data set. For example, if we do the kidney transplant, we only use the kidney transplant data to input in our agent system. And how does the agent system handle the batch effects? I think I can put more words on how to use this agent system to process the data or how they take the data in. So as we know, we built the agent system from our large language model, but we didn't directly use the large language model to process the raw data. For example, we didn't, we used the GPT-4. We can give more details of the implementation. We used the GPT-4 provided by the UCSF API. So we didn't input the raw data like mRNA profiling. We didn't input this kind of data and also lab test directly into the GPT-4, but we use GPT-4 as a core brain to process the results. For example, it can use a machine learning model to process this raw data. For example, we use the light GBM to process this raw data, and then we will return to the results to the GPT-4 that, okay, here we use the mRNA profiling and the lab test as the input, and we have this kind of results. And then the large language model will determine whether we should add more features, well, whether we should process more data into this input. So that's the way we process the data. Does that answer the question?
Okay, maybe we can move on to the next questions and can get back. So the next question is, how do you know if these studies also contain the type of rejection like EMBR, TCMR, or quality, etc.?
Okay, how do you know if this does all contain type of rejection? Yeah. So some of the study they have more specific of the rejection type. For example, the acute rejection. Some study they haven't record that kind of information. So currently the way we process this kind of information is we didn't classify it into a very specific like acute or a long-term rejection. So we put it in a general category of rejection and non-rejection. And in some study, they only have the data like rejection and non-rejection. Yeah.
So the next question, could you tell us like if these input features were collected prior to the transplantation or also after
Oh
transplantation?
yeah, so that's a good one. So some of the features they are collected before the transplant, and some are collected after the transplant, but they are before the rejection. So it from a there's a mixture between the pre-transplant and the after transplant of the rejection. Yeah.
Okay. Can you elaborate on how missing data and class imbalance was handled, especially for the training and validation split?
Okay, yeah. So, missing data, we using some traditional and commonly used input method to input the missing data. For example, input the zero or input the average value by the close data points. And for the class imbalance, because from our experiments, we find that the data imbalance is not that severe. So we didn't have a specific process on the class imbalance. Yeah, and the train and the validation split, yeah, it's a five-fold cross validation. So we divide the train and the testing in like a five rounds, like yeah. So it's a cross validation, k-fold cross validation.
All right. So the next question is, can you explain how to train the two AI experts to make them talk and discuss?
Oh yeah, so how to train the two experts as to make them talk and discussion. Yeah. So because our agent system is built from two large language models, so we can let two large language models talk to each other. It's like a large language model A can output some sentences, output some answer, and then we can input this answer to another large language model so that they can talk to each other. So the core of the agent system is the large language model. Yeah.
So do you plan to use any independent data set to validate your results?
Yeah, we have a plan for that. We have a plan for that. For example, for kidney transplant, we can use an external and independent data set to evaluate it. Yeah.
Next question, how do you ensure that the biomarkers you identified are not learned by the LLMs from pre-existing knowledge?
How do you ensure that the biomarkers you identify are not learned by the from pre-exist? Okay, so yeah, I think the way we use large language model is that we want to use it. We actually want to use the large language model's pre-existing knowledge. We want to use that. For example, the large language model may know the sodium. It has a very strong relation with the liver rejection. So we want to use that kind of knowledge. And that is why we can use the large language model to optimize our input features. But we also want the large language model to have some more exploration than this kind of exploitation of the pre-existing knowledge. So for the exploration, I think we can have more optimization on the prompt that we use for the large language model. We can tell the large language model that, okay, you can explore more to the unknown knowledge, to explore more that the biomarkers that seems didn't has a relation with the rejection. So that it can be achieved by the prompt learning or this kind of related technique. Yeah, we can have a balance between the exploration and the exploitation. We need the pre-existing knowledge from the large language model, actually.
So there's a continuation to that follow-up question also. What's the computational load to run the framework you presented?
Yeah, it then has a very large computational load because we use the API of the GPT-4. So for we don't. But if you use the local large language model on your on the local server or the local computer, we need large computational load. But if we use the API provided by, for example, by UCSF private GPT-4 API, we didn't need many computational load, and it has a very quick um, runtime for the framework.
And if I may say, I think you have used the UCSF Winton server.
Yeah, Windows server. Yeah, on the Windows server.
To the next question, how do you choose to reconcile disagreements between the two agents? For example, if the ML agent indicates a feature is very predictive of rejection, but the clinical agent feels it is not biological relevant.
Yeah, that's a very good question. So, there's this kind of situation where happens, I cannot say frequently, but sometimes it will happens. So the way we solve this problem is, we solve it in an engineering way. So we will set up a maximum discussion round between the machine learning agent and our clinical agent. So as long as they achieve this kind of maximum discussion round, if they still hasn't reached an agreement, we will run it again. So it's more like an engineering solvement of this kind of issues. Yeah.
Okay, so next, very nice work. You mentioned the use of LGBM to process raw data, and then the same is used for prediction. Does it take into account data that was withheld from training for evaluation? And what is the minimum data set required for robust prediction?
Yeah. We use it. So it didn't has a like a data leakage problem because we use it. For example, only use the when we do training and when we do the optimization, we only use the training data. And for the testing data, we will just use the already trained large language model and using the same set of input features. And what is the maximum data set required for the minimum data set? So okay, so minimum data sets. So for the data set scale, because we didn't have used any deep learning models to optimize, and because large GBM like GPT has already performed a very good performance to use as a base model. So we didn't have a very large requirement, high requirement for the scale of the data. So small data set is also okay. So we can find from our previous slides that these studies they didn't have, for example, thousands of patients or hundreds of thousands of patients. We only have several hundreds of patients, but that's good enough to train and testing the model and evaluate the model. Yeah.
Now to the next, how does the tool calling work in terms of processing time and data format output?
Oh yeah, so I think that's by coding and engineering implementation. So for the data format output, you can make sure that the model by prompt learning, you can like make sure the model outperforms and write like in JSON file. Sometimes that can help with the large language model to process and to uh, Python code to process. Yeah, that is kind of achieved by the engineering by more complicated coding and better prompt learning to the large language model.
Okay, so there is a question about video recording of this presentation. Yes, we will be, this session is recorded and we will be sending all the recording to all the registrants. So soon you will be receiving the recordings. Okay, there's one other question coming up. If your system will have access to openly available information for improving biomarker implementation, how will you protect the donor and patient in the case of kidney transplant, PII used for specific case decision making?
If your system will have access to public available information for improvement, how will you protect the donor and the patient in the case of being used for specific case decision making? Oh yeah, that's a very good question. So that is the question related to in the large language model era, how how we can protect the input features, especially in the biomedicine and healthcare domain. So I think one solution. So currently we use because we use the like a UCSF API of the GPT-4, so it has a good protection of the like this kind of privacy. But if for other kind of circumstances, we can use the local large language model. We can have, for example, we can have the large language model or foundation model in the local hospitals, and then they can use the local version. For the local version, they can protect they can direct input this kind of patient's data into their local versions, large language model and the foundation model. Yeah.
All right, I think we are coming to the end. All the questions got answered live, and now we have roughly about 4 minutes left. Probably I'll just take a moment for some quick announcements. Thank you, David.
Thank you.
Right, so let me share. There you go. All right, so yes, so we are excited about and I would like to just announce that some exciting upcoming event. So on Thursday, June 12th, from 8 a.m. to 11:30 a.m. Pacific time, we are hosting a virtual symposium titled Role of AI in Immunology, Advancing Scientific Frontiers. And this event is a part of the Focus Research and Education Virtual Symposia series. And we will be focusing on artificial intelligence, how it's transforming the way we understand and apply immunological insights. It's organized by ImmPort, and we are expecting a diverse audience from clinicians and researchers to data scientists, industry folks, and so we don't expect any prior knowledge. It's more a session of invited talks and a demo of how to access ImmPort data sets or also access the AI-ready data sets on ImmPort. So please feel free to scan the QR code on the slide to register or share it with the colleagues who might be interested. We hope to see many of you there. And if you want to stay connected with ImmPort for upcoming webinars and explore new data released through our mailing list, newsletter, visiting our website, follow us on social media. Here are the links. We also have the QR codes, which will take you to the ImmPort registration page. We have UCS ImmPort tutorials developed by the researchers. That's also on the ImmPort website, ImmPort AI-ready datasets, and many more. So with that, I think we're going to give you about 2 minutes back. Thank you all for joining today's webinar, and we appreciate your time and interest in learning more about ImmPort. Thank you.